students and professionals could come and get the training that they need to go work day one at a major company like a Genentech, like a Merck, like a GSK or Pfizer or Janssen or Johnson & Johnson. And as I cross through this space, you can see here, students such as yourselves, the students who are in master's program right now, have the opportunity to work on what I call this next generation of manufacturing. If you've ever been to, you guys are all chemical engineers, you guys have studied probably reactor design and chromatography systems and all those separation processes, all that good stuff. Well, in biopharma, we do have some of those larger facilities, but if you ask me where your generation is going to be, you guys are going to be working on this scale, right? So one thing is these wonderful monoclonal antibodies that we're producing, some of these new modalities need much, much smaller volumes. And so we're able to do this now. Instead of building a big stainless steel tank that you see in your textbooks, what we're able to do now is, if I scroll a little bit closer, if you guys could see it, now we're able to do things on a much more process intensified scale. So we have these plastic bags that we insert. Now, instead of having to clean a reactor, re-sanitize it after we grow our cells and produce our protein, now we just stick a bag in, put the cells in there, and toss it out and go to the next molecule, the next batch. That allows us to process things a lot quicker. If you guys ever done these simulations, that time is money, time is to the clinic is money, time to the clinic is meaningful for the patient. So if we're able to do things faster, more efficiently, obviously that has a benefit for the patient, obviously, and for the company. And so what we do here is train students and train industry professionals who come to us on these processes, whether it is how to design these processes, how to operate this reactor properly in a GMP setting. What does it mean to work in a regulated environment? How do you build quality into your processes? All of that's covered in some of our programs. And again, we do this in a real life setting. So you get to work with these large reactors, these big mixing tanks that are used to do certain operations, which I'll talk about in a moment. If we go into the next space here, if you guys have taken, I'm not sure if we have any seniors or juniors, you guys have all taken hopefully heat and mass transfer, maybe your separations courses. So you're familiar with filtration and chromatography. So if you guys had, maybe you have internships, but you might have seen this, but if not, I'll show you guys. We have these pretty large scale chromatography skids. In the industry, these chromatography systems, which are used to design to separate the proteins that we want from the proteins that we don't want, those columns could be 60 centimeters, 2 meters in diameter, right? So how do you train somebody on something that big, which costs a lot of money to operate, and there's no school in the country because has the resources to do that, to my knowledge. But so we invest in this set of equipment to give you guys this kind of exposure, how to pack the column, how do you evaluate the column, how do you run this, how do you generate the proper data that you need to go into a filing for FDA submission. All that is covered in types of programs from a practical standpoint, and obviously from a more theoretical approach as well. But that's just MABs. That's just the beginning of the journey. And the reason why we focus a lot on monoclonal antibodies, I should say, when I say MAB, I mean monoclonal antibodies. The reason why we focus on that so much is because if you guys go directly right now to work at these major companies, I could guarantee you, your guys are going to be working on producing some sort of monoclonal antibody at these major companies, because that's the predominant molecule right now in what's called the large molecule space. Now, our facility, obviously you guys are familiar with pharmaceuticals possibly. That also covers small molecules, but we don't do any of that. We don't do a small molecule. We don't do Tylenols, Advils. The only thing we focus on are these what are called low volume, high value molecules. So monoclonal antibodies, replacement proteins. And in addition, we also have a lot of projects in this next generation of therapeutics, which are cell and gene therapy. So you might have heard about cell therapy products. You might have heard of gene therapy in the news or from your professors. We also cover training and education and research in that space as well. So I'm just taking you through some of our laboratory spaces as we go through the, forgive me if it's a little bit jumpy. In addition to all that, in addition to what we call process development, manufacturing, training, and operation, we also have an excellent program, piece of a program that's covering analytical. So there's my counterpart who's another professor and also an analytical chemist. He covers what's called regulatory quality and analytics, which is a key feature for any student, any entry-level employee to understand the regulatory, because we are one of the most regulated industries in the world. How does that play into what we do every day in our daily jobs? Quality, what does that actually mean? When I say quality, and analytics are our eyes for the process. How do we know how do we know when we operate that bioreactor? How do we know we've produced enough protein? How do we know that it's the correct protein? all that is covered on analytics, and that's also part of our program that you guys could get if you guys choose to come to Jefferson. It's one more laboratory space. And the cool thing about this space here is, I mean, I introduced myself as both as operations director and the professor in the engineering school. The cool thing about this is that it's very, very industry focused. Everything I do, everything our staff does, and the people that work under me, everything we do is industry focused, right? Now, that doesn't mean we have an academic spin to it. We have PhD programs, we have obviously master's programs, we have certificates, we do all that too. But the value that we see is not necessarily what you get in the textbook, which is good. No, I'm not saying I'm not knocking it, it's not good. But you also need that other piece of the applied part. What do I need to do? What do I need to know? And how do I do it if I want to get a job in these companies, and not just get a job, but also be successful and be a fast riser through these companies? If I could, I should actually invited one of my previous students. He just started a job at GSK, which is a major biopharmaceutical company. And he was just telling me the other day, I was talking to him, that he's been there, he just graduated, he's been there about 3 months, and he's already been assigned a lead, which is unheard of at this company, for somebody to be that new, to be assigned the lead on a project. So that's sort of just a testament to what kinds of opportunities, what kind of training you guys get at Jefferson. So you guys get to, because we're so industry facing, we get a lot of projects from companies, large and small. So last summer, we had projects from GSK, we have projects from other small companies in cell therapy space. And the students, like you guys, got to work in the laboratory, working on projects, working with the scientists at the other end, producing data, generating reports. And these are actually projects that the company actually valued. So those are other cool things that you guys could do here besides just the classwork. But we love to get you guys engaged on these industry projects as well, along with the rest of our staff. So you saw that big reactor that I showed you earlier. These are some of the smaller reactors that we have. So you don't want to run those large reactors in industry. If you're running an actual product in that reactor, you're talking about millions of dollars worth of value for that reactor. And that doesn't even include the actual consumables, the cost of goods for running that, and the fact that it might take about 4 weeks to get from the vial, which is where you start, all the way to that, even a 200-liter scale reactor. So as you can imagine, if you get a contamination event, you see how the companies are really stressed out about making sure that you guys are trained before you start working in this space. And so that's what we're here to address. That's my only job. I joke with some of my students that my job is here to serve you guys. My job is to get you guys a job, and that's what I take seriously. That's what hopefully you guys get from this presentation. So this is just, again, this is I'll just take you around a little bit more. This is what we call upstream development lab, where you have these small reactors. Many have seen this scale before. If not, again, if you have a chance to visit, you guys could see it in person. And if I just pop back out, we also are currently planning expansion to the first floor of our facility. So by the time if you guys choose to come next year, we'll be well on our way to constructing that space. And that's going to be even more capabilities. So you saw in that image I showed you guys, we have what's called a 200-liter scale single use. That's all. The bags are called single use bags. Those bags can get up to at least 5,000 liters in size. We'll have, actually I do have on site right currently a 1,000-liter bag. So that's the scale we're talking about for you guys to get training on. And we also, all the other necessary equipment to operate at that scale. But in addition, the thing that's even cooler is that we're also going to have more spaces for the cell therapy, the gene therapy, the advanced vaccines, the viral vector manufacturing, all those buzzwords you guys might be hearing about. We're also building more capability. Even though we do it now, we'll have even more capability in the near future. So I just wanted to leave you guys with that, to show you what we're doing here, how we're growing, expanding, and all the wonderful opportunities that exist for you guys at JIB. So I'll stop there for the tour, but also going to show you some slides with you guys, if you don't mind. All right, so give you guys a little bit of context after you've seen that presentation. All right, so again, our program it's called Biopharmaceutical Process Engineering. And I really want to hone in on a couple key concepts here, because I told you I did biomedical. I think you guys are BMES here. I was a member of BMS when I was in graduate school and undergraduate. SWE, my wife is also an engineer in a pharmaceutical company. She works as a process developer. She was participating in SWE. So both of your organizations are very near and dear to my heart. This will give you a couple high-level things about us. This is really don't want to bore you too much. But again, this is our mission, obviously, here is to deliver and educate the next generation of skilled workforce. For the region, actually nationally now, when I wrote this we were focused primarily on Philadelphia, but now we've expanded. We have partnerships with schools in California, as far away as California. So we are a very national focused organization, and we try to be affordable. We're working on we have a wonderful scholarships for students, but we're also working on procuring more. And again, these scholarships are for students supported by industry. So you would have a named scholarship by, one of them is from a company called Paul, which makes one of the biggest players in filtration technology for biopharma. And we have others that are coming along. So you have lots of opportunities for financial support if you guys need it. We're very accessible, in terms of giving you guys opportunities, but also even myself, like I offer my time to all the students. I'm not running a secret lab somewhere. The only thing I do is really focus on you guys and this facility. And we want to build awareness for biopharma. I can't tell you guys how many times I've talked to senior people in the industry and they say to me the same thing. The students are graduating with very little exposure to what we do, right? And I'll show you guys why that's a problem as we move through this next century, which is really the century of biologics, century of biopharmaceuticals. Your careers will be dominated by your contributions to the next generations of medicines that are going to help improve patient lives, even save people's lives, even cure people in some instances. And so my job here is what little I can do is to expose you guys to, hey, when you guys graduate, and as a chemical engineer, I worked at a chemical company for sometimes. I know that space very well. I was making polymers, plastic polymers. I've done the hardcore chemical engineering myself. And I'm telling you guys, there's not to knock them, there's wonderful opportunities there, but that's not the only thing. And for you guys going into more of the life science aspects, you really hone in on what is industry really looking for, what skill sets are these people in the industry looking for me to have when I graduate, and how can I better enable that so I can have a more a fulfilling career, right? And I'm going to throw this is really some more information about JIBs. I want to skip over this so you guys can understand that. Really, I just want to touch upon, I'll start here, is that what is the biopharmaceutical industry, right? So, what is it? So to me, the biopharmaceutical industry is really to be honest, I have a lot of physician friends, but really, this is the underpinning of modern medicine, right? Where would medicine be without insulin, without Herceptin, without Keytruda, all these molecules, these blockbuster molecules that are here to address breast cancer or melanoma or insulin, diabetes, I should say. All of that is captured in what I call biopharmaceutical vaccines. We looked at the COVID vaccine, right? So that's the contributions that biopharmaceuticals has made to our daily lives, helped us to get over a pandemic, helped us to help our family members get over certain diseases, right? That's what we do, right? We save millions of patients at a time. If you look at the contribution to the U.S. economy, this is probably a few years old now, but we're talking trillions of dollars of contribution directly to the U.S. economy. This is a huge industry. If you look at the top companies, I can name a few of them off the top of my head: Roche, Genentech, Pfizer, Janssen, GSK, Merck. Right, these are some of the biggest Fortune 500 companies. And it's really a shame to me that sometimes students don't really realize that they're there and they're looking for you guys. They're desperately coming. They knock on my door every day. It's like, Cameron, where are these students coming from? I'm like, I'm trying to get you guys interested, but this is, and if maybe you guys find it by happenstance, but I want to sort of fix that. I want you guys to be directly trained to work at these major companies and contribute from day one, rather than having to come back to me in a year when these companies send you guys back to do more training, which is what happens currently. That's what we do a lot of. If you look at the number of jobs directly tied to biomanufacturing, it's probably going up actually during the pandemic, but we have about 1 million jobs directly tied to biomanufacturing. These are the companies that actually produce the medicines. But if you look at all the jobs that support that industry, we're talking about maybe 4 or 5 million jobs in total, going from all the sales and the vendor, the technology providers and the vendors and all the other the logistical arms that these companies manage. All that has to be goes to support these industries. So there's lots of jobs. If you don't want to go directly into making the medicines, you can obviously have opportunities in either making equipment, making media, making the bags. All those things are also part of the biopharmaceutical industry. The average salaries are very competitive, especially for engineers like yourselves. And as you accumulate a master's and PhD or have more work experience, obviously that grows. The number of the interesting thing for me, and I try to tell students this, I say really bluntly, I tell students to follow the money, right? Now I know you guys are in Texas, but you guys are a BMES. So you guys have sort of already sort of adopted some of the philosophies I'm about to say. But if you guys think about it, it's not a common occurrence. I'll say this: it's not a common occurrence that we build another chemical processing facility or refinery. Those things are rare, even though we do sometimes, but that's very rare nowadays. If you look at the companies, even these companies I mentioned before, they're outsourcing or divesting a lot of their small molecules because that's being taken over by generics from India and China. Where you see investment in the future is in three areas. These monoclonal antibodies that people are producing, that these companies are producing, they're really heavily into not just mAbs, but also other types of fusion proteins and other complex molecules. That's where they're investing most of their money. And they're also investing heavily in cell therapy companies and gene therapy. That's starting to kick off now. So I'll give you an example. I previously worked in California, and I did work closely with a company called Kite Pharma, which is one of the first cell therapy, first companies with an approved cell therapy product. And I was there when they were purchased by Gilead. Gilead invested about I think it was $13 billion to buy that one company, right? So that's why I tell students, follow the money. If the companies are putting that much money and that much resources in a product or a molecule, that's where you want to be working, right? That's where the investment is, right? So, unfortunately, we had the issues with the, pandemic and everything, but if you look at our industry, biopharmaceuticals, we actually grew, whereas a lot of industries, unfortunately, had family members impacted other industries. But in our space, they're actually increased the number of jobs. The number of projects that we had actually increased. And that goes to say is that we are very insulated from economic downturns for the most part. Now, obviously, here and there, some things happen, but for the most part, for the last 40, 50 years, biopharmaceuticals has been growing and is actually anticipated to grow even more so over the next probably 30 to 40 years. So by the time you guys finish your careers, you guys will probably have spent your entire careers working in biopharmaceuticals, which is a great opportunity. So the reason I say that is because if you look at the reason why you guys will have those opportunities, because if we look at the clinical development pipeline, there's thousands and thousands of molecules that have to get developed. Now, all of them might not make it, but you still have to do the work to see because you don't know what's going to fail, what's going to succeed. So you still have to do the processes. You still have to take them through that journey until they fail. So there's lots of work for you guys to do. There's plenty of what's called first-in-class medicine. So we're constantly striving for new indications, treating new patients, finding better ways to treat older diseases. So that's something that is an attractive opportunity for students to work on something fresh and new. A lot of these molecules are in what's called oncology, cancer medications, heart disease. The next big challenge, I think, in my opinion, is neurological diseases like Alzheimer's that still hasn't been figured out. I mean, we used to think that we understood how Alzheimer's progresses, but if you ask people in the industry, they will tell their academic colleagues that, hey, maybe the amyloid beta or the tau protein hypothesis may not be correct because we've been developing molecules for these targets for years and it hasn't worked. And this is like hundreds of clinical trials. So that's something, if you guys are up to the challenge, that's something for your generation to figure out. Are the ways we can therapeutically address these diseases like Alzheimer's and develop those medications, which obviously will help hundreds of thousands, if not millions of patients in the future. I always like this slide. This is interesting, just to give you guys some context. And every time I give this to a talk to an engineer, I like to ask you guys a few questions. So if you guys want to just jump in here, so I'm not talking the whole entire time. If you guys have any guesses as to what is wrong with this, what's afflicting this child in this picture on the left? Do anybody have any guesses? And you can just speak up. You don't have to put it in the chat. You can just shout it out. Any guesses? There's no wrong answers. Don't be shy.
Is it a Spanish flu?
Somebody says Spanish flu. Dorothy says Spanish flu. Not the Spanish flu, but that's a good guess. Anybody else? Thank you, Dorothy, for being so brave.
I say diabetes.
Diabetes. Did you cheat, Omar?
No.
Yes, that child has diabetes. So this is in 1920. And actually, think about it, this is really only 100 years ago, right? This is 100 years ago. 100 years ago, this child would have died, right? Now, if you look at the picture on the right, you see the same child about 2 months later, he's looks more obviously more nourished. His cheeks are pudgy. I have a small son, so my son has the same pudgy cheeks, right? He looks more nourished. So this is a testament to the power of what I call one of the first biotherapeutics, and this is obviously the case of insulin. So this is one of the first demonstrations that you can give a patient insulin and helps them regulate their glucose levels. And obviously this boy was able to recover. Obviously you have to take insulin for the rest of his life, but he's at least able to recover from the terrible state he was in before, and only a few months earlier. So really, again, I tell students too that why do I do this? Why have I done this in my career? Why does the staff at JIB do this? Well, obviously, it's a great career. Those are all great, but at the end of the day, when you see this is why you do it, right? I'm helping potentially save somebody's life, improve somebody's life, and really you go home at the end of the day with however much stress you have, you can always go home and say, if you own a molecule that's on the market, you can say, today I helped somebody improve their life a little bit today. And so that's what at least I take home. And this kind of story actually just hammers that home a little bit more. But that's cool. That's the science of it. That's what the doctors care about, right? They give the medicine to a patient. The person, he or she gets better. What I like to say is, from an engineering perspective and the bioprocessing perspective, to me, my question is how and how much. Okay. So in those days, in a company called Eli Lilly, you might be familiar with them. Eli Lilly was the company that started commercially manufacturing insulin. The way they used to do it was really, really, really crude. Now, from the perspective we have today. So they used to go around, if you go visit Lilly's campus, you can see that they have these huge vats where they would collect spare pig parts, and they would grind them up and extract the insulin. If you see here on the bottom of my screen here, you see the different vials, the different stages of purification as we get more and more pure product. To be honest, actually, I'll touch upon this why. We could never give somebody that insulin today. The FDA would never, ever allow you to do that. But I'll talk about that why in a second. But here, this is, if you look at the amount of pig parts it takes to make 1 8-ounce bottle of insulin, it took about 2 tons of pig parts. That's a lot of pig parts, right? So, and it was a 4-step process. You lost a lot of insulin along the way. So then they would go around with huge trucks and go collect the pig parts. But then they did a calculation, right? They saw that the number of people, especially with type 2 diabetes, was going up over the decades, and the number of pigs was probably essentially a fixed value, right? So they said to me, will there be enough pigs for us to supply the market demands, in this case, the patient population, for insulin, right? For extracted insulin. And so they said to themselves, the answer is no. So I want to ask you guys a question, right? So let's do a calculation. Let's take out a piece of paper and pen. Okay. So if we estimate today, and this is a couple years old, but if we have the most recent data, it's actually gone up probably 600 million. The total patient population, people who have type 1 or type 2 diabetes, is about 500 million people, right? That's a staggering amount of people, okay? And if it took, I told you it took 2 tons of pig parts to make 8 ounces of insulin. So if we do some math here, if we say it takes about 10 pounds of pig pancreases per year per patient, which is basically the dosage, and then we estimate the average weight of a pancreas per pig, we get that it takes about 15 billion pigs per year to satisfy the demands for 500 million patients. 15 billion pigs. So Omar, since I can only see you right now on my screen, can you guess how many pigs there are in the world?
I would say like 50 billion.
He said 50 million. Okay.
No, 50 billion. Would it be?
Oh, 50 billion. Okay. 50 billion. Anybody have another guess? What about Mary? What about Mary? What do you think?
Like like 2 million. I don't know.
Okay. So you guys are 50 billion is too high, 50 million is too low. All right. So here we have, if you add up all the pigs farmed worldwide, right? China comes in number 1. They have about 440 million pigs. But here we have If you add up all of them, the US actually only has 73 million pigs. We only have about 630 million pigs in the world as of 2018, right? So I can't imagine it's gone up that much over the last 2 years, 4 years, right? So that's a problem, right? We need 15 billion and we only have about 630. And obviously we're not going to use every single pig on the planet just to make insulin, okay? So what do you think they did? They might have a guess.
Is it recombinant DNA technology?
Recombinant DNA technology, yes. Do you know the story about that? I'll tell you, I could tell you the story. So yes, so back in the late 70s, the Eli Lilly did this calculation. So they put out a recall, they said, or a project request, and he said, are there any smart scientists out there that can figure out how to produce insulin in a different way, right? They didn't specifically state how. So the recombinant DNA technology is what came out. That was the technology that won at the end of the day. But yeah, there's a couple. If I remember the story correctly, is this a couple people that they weren't necessarily that young, but they were, pretty junior scientists. One of, I think they're probably in their 30s or 40s, if I remember correctly. And they that and another group also were the ones that were credited at least with demonstrating that you can take the human gene for insulin, insert it into a plasmid, put that into E. coli, and express it, and it will produce insulin. Right now, that sounds all cool. This recombinant DNA technology. To be honest, actually, that's the old way we're gonna do things, and we do things similarly, but a little bit more efficiently today. We don't produce things mostly in E. coli. We actually use a different cell line. But when they figured out how to do that, that changed the game. That was what we call the Big Bang. So in 1983, that's when insulin was first commercialized, available in the market. And obviously, today, to be honest, Lilly is still making insulin the same way they were in 1983. It's almost 40 years ago. 30, 40 years ago. They're still making insulin the same way they did before. And that's because of the nature of how regulated we are. You really can't change the process. But that's what we call the Big Bang. And this is a funny story. One of the groups that won the contest actually started a company, and that company was actually Genentech, right? So Genentech, which is probably actually the biggest, largest biopharmaceutical company in the world, was started out of this project call, and they dominate the biopharmaceutical space. They're one of the leaders, the global leaders in this space. So if you look at Lilly, so they were able to make insulin recombinantly. You take your if you have diabetes, you take your insulin shot multiple times a day. That sounds great, but now we've actually moved on. So companies actually like Lilly here have moved on to producing new molecules. And if you look this up, you'll find this molecule. It's probably about a $2 billion yield drug now. This is a molecule called Trulicity. So instead of giving the patient insulin every 3 or 4 or 5 times a day, this molecule here is a fusion protein with a molecule that helps you regulate your insulin levels attached to the back half of an antibody that allows it to make it a little bit larger so it stays in your system a little bit longer. And so what that does is now instead of taking multiple times a day, now I could take this molecule once a week. So from a patient perspective, right, you can imagine if I have people in my family who have diabetes, it's really a challenge to get people to properly regulate their glucose levels. And because they don't, they have their disease progresses in the negative direction. Here, imagine if a person only has to take a subcutaneous, sorry, I'm using big words here, take a shot under the skin every once a week. Imagine the improved compliance. Now, your grandmother, your mother, your uncle, your brother, your sister, whoever it is, if they take it once a week, they will take it once a week. They won't forget. It helps them regulate their insulin levels a little bit better. And obviously, that's a big improvement on that patient's life. So that's what we're doing now. That molecule is already on the market. It's already being sold. It's a very successful molecule for Lilly. So you guys, that's the generation. This is the current generation of technology we're working on now. These large, complex proteins is what you guys will be working on if you go to these companies. Now, the next generation, where you guys will also make contributions, is now moving towards what's called cell therapy, where we're either taking the cells from immune cells from a patient and reprogramming them and having them target cancer, or we're giving patients gene therapies. We're moving to next generation vaccines, like the Pfizer, Moderna vaccines. Those are what are called next generation vaccines. We need talented engineers like yourselves to make these things, right? There's a big demand for this. People need medications. And the question for you guys is, are you up to the challenge? Can you help the next generation? Can you be part of the next generation, I should say, that's going to make these next generation of molecules? And so where we are today with cell and gene therapy is actually where we were here with this patient extracting insulin from a pancreas, right? If you look at it from a processing perspective, the technology looks cool, but the actual fundamentals of the process are very rudimentary, and we need engineers like yourself to improve them, to get them to the point where insulin and where these fusion proteins are today. So that's your challenge. This is quickly here. I'm just going to go over this quickly, but this is just a little bit about JIB. We were featured in this article from a few years ago. If you look at all the major biopharmaceutical clusters in the United States, from San Francisco to Boston to North Jersey to North Carolina, Philadelphia ranks around 5 or 6, depending on the year who's calculating it. We're on 5 or 6 biopharma hubs. And I like this because we were shamelessly put this in here because we were featured in this article talking about how this is part of that region, part of the analysis of why we're a hub is because JIB is there to train the next generation of workforce. So I just a shameless plug there. But here is, again, more information about our what's a cluster here, what do you need to have. So it's not just the science. So it was wonderful. I have wonderful colleagues who are professors and researchers. And we need that. That's part of the foundation for the science. That's the science behind what we do. We need those people. We need those discoveries. But the discoveries don't automatically mean a commercialized product, right? For every 10,000 molecules in discovery, maybe 10 of them ever reach the market, and that's within 10 years. So what happens between that period? Well, those other 9,000 or so molecules all failed, and for one form or another, for some reason, they all failed, right? So in your careers, when you're at these companies, you will most of the time you only work on one or two molecules that ever make it to market, right? If you've been there for 30, 40, maybe 20, 30 years. But the ones that do are very valuable. That doesn't diminish anything about what you produce. It's just that's the nature of the beast. We don't know when you put something in a human, you don't know if it's going to work or not. You don't know if it's safe until you put it in a human being. Everything can work in a mouse, but very few things work in a human. Okay, so that's the science part. So how do we get from the discovery, the benchtop, the scientist that's maybe discovered a new pathway, a new molecule that could potentially target a pathway? How do we get from there to a Herceptin or insulin? Well, that's what you guys do. That's what we train you guys how to do. That's what's called process development, product development, and that's what's the missing piece between the science and the physician, right? And all that goes into this piece here of these clusters. That's why these clusters are there, and what and how we put all this stuff together. Again, this is more information about Philadelphia region, about the number of investigational new drugs in this area. Someone skip over this slide. Just move on to the more interesting stuff. So I tell students, that's what I said, follow the money. If you ask me where you should focus your careers, focus on where there's areas of investments. So there's things like you might be working on, things like tissue engineering. I can tell you right now, tissue engineering is great. I have some friends that did this when they're doing their PhDs. But if you ask me from a commercial standpoint, we're talking about, from a real commercial standpoint, we're talking maybe 20, 25 years before it becomes a real valid, widely approved technology for treating a host of diseases. Where companies are investing is, like I said, in monoclonal antibodies, complex proteins, and then some cell and gene therapy. Right. So if you look at the pipeline of molecules, this is what molecules are in phase 1, which is the first in human. You have phase 2, which is an extended or a more advanced study in the clinic. You have phase 3. BLA is when you actually submit your molecule to be approved, to licensed, to get it approved to sell on the market, and obviously market. We have 50% of what's called these large molecules are antibodies, right? Then 75, 80%, 90% of the molecules are in development of monoclonal antibodies. So if you ask me, like I said, if you ask me, what am I going to be working on? What do I need to study? You need to study antibodies. This tells you what you need to study, okay? So if you guys, again, that's what we focus on here at JIB. And this is actually a couple years old. So you can imagine this is probably even escalated even more, okay? Now I focus on the antibodies, but there's a whole portfolio, a whole series of molecules that you guys can work on, or will have opportunities to work on besides just antibodies. So there's also therapeutic proteins, there's small peptides, there's what they call these bispecific proteins, where combining half of one antibody and half of another. Actually, there's also what are called tri-specific, so they put 3 of these together. So these are very challenging. So the science is cool, but from a chemical engineering standpoint, our question is, how do we make this at scale, right? If I need to treat 1 million patients, I need to be able to make this at large volumes consistently with high quality, right? And these molecules are very, very sensitive, right? So that makes it more of a challenge for us. People are working now, you're just starting to see these more widely approved, what is these antibody drug conjugates. So you have your monoclonal antibody, and you could decorate this with chemotherapy agents. So now you could directly deliver drugs to a tumor site. Those are very complicated to make at scale and consistently. Fusion proteins, they seem simple, you can make them in the lab, but when you need to make them in that 200 liter scale reactor, that's a different challenge. How do we purify it? How do we separate it to make sure we have a high quality, high purity molecule? That's the question that you guys have to answer. For CAR T and other cell therapy products, now it's even worse because the molecules, it's not just a protein, it's a whole entire cell. And what does it really mean? What cells do I actually need to give the patients? And that's the open question right now. It's one of the major challenges for cell therapy, is really characterizing what is a high quality cell product. We don't know that yet. For the same thing for gene therapy and viruses, the same complexity. What does it really mean to have a high quality viral vector, a high quality vaccine, a high quality CAR T product, a high quality fusion protein? Those are the questions that you guys have to answer. And once you answer it, then you have to be able to make it consistently, because once you get it to the market, without some minor exceptions, you cannot change that process. So whatever you go commit to, you're committing yourself to not just the molecule, but you're committing yourself to the process to make that molecule. That's what you're telling the regulatory agencies. So the challenge for you guys is really defining that process and making sure it's going to work, not tomorrow, not a year from now, but also 40 years from now, like insulin is, because people are going to still need this medication years from now, right? That's, to me, that's the exciting part. You get to make a decision that's going to last well beyond your career, right? Some of the challenges here, like I said, about the complexity, right? So for small molecules like aspirin, we can make these in a pot. Like you guys have taken your reaction kinetics, you guys have taken organic chemistry, so we can synthesize these molecules from scratch for the most part. But that's like putting together a bicycle, right? Then we realized that we can't chemically synthesize large proteins. So we had cells do them. So we had insulins, we had the human growth hormones, much larger, more complex, but still they were like a small car here. Now, if we talk about the monoclonal antibodies, the biospecifics, the fusion proteins, that's like putting together a jet, right? These molecules not only have different structural features, but they have what are called post-translational modifications and other modifications to their surface that could potentially impact what's called safety or the efficacy of the molecule. And how do you characterize that? How do you run your process to make sure that I'm getting the right window, the exact right monoclonal antibody that I need to give the patient? And that, again, that's what we teach you guys here at JIB. This molecule that's spinning to the right here, this is a molecule called Keytruda that I mentioned before. This molecule is produced by Merck, and it's estimated to make about $20 to $30 billion a year. I said again, $20 to $30 billion a year is for melanoma and a host of other cancers that have a particular marker. And if it goes as indicated, as expected, Merck's going to be a very rich company. So, and there's other companies that have some challenges to that molecule in the space too. But again, this is, to me, is one of the next blockbusters. This is what's really going to change cancer treatment over the next decade or so. And then lastly, we have cell therapy. Again, I told you about the challenges here. If you can imagine producing an antibody is a challenge, wait till you get a load to what's happening with cell therapy and the complexity around understanding how cells express certain markers on their surface and really quantifying that. That's a major challenge right now for the industry. Here's just a sample. This is what we call the platform process for monoclonal antibodies. So really, we've landed on through years, about 30, 40 years, we figured out how to make monoclonal antibodies consistently at large scales. And here's just an example of the process that we teach you. We go through each unit operation, what it means, from an industry perspective, we give you real life industry examples, not stuff that we just come up with a whole cloth. We actually give you guys examples that from real life examples that we had in our careers that helps you guys understand how the challenges for making these molecules and what you need to learn when you go into the space. Again, I'll just end here about quick. And I'll stop here and I'll open up for questions. Again, this is more information about Thomas Jefferson University, a very large school, 10 colleges, 3 schools, wonderful history in life science and healthcare. Our engineering school is strong in engineering, design, mechanical engineering, and obviously biopharmaceutical engineering. Lots of great stuff here about the university. And we're actually located in multiple campuses. Again, like I said, we're located a little bit outside of Philadelphia. This is some more information about the university, which you guys can find online. Just quickly about our master's program, and I will stop here. We actually do a calendar year master's program. So we go from September to September or September to August. Our first semester, we take life science and engineering students. So if you guys come here, you'll have colleagues who have life science backgrounds like biology, biochemistry, and then we even have people who are mathematicians. And you guys, if you're a life science person with very little engineering background, we ramp you up on certain courses. If you're an engineer with no life science background, we give you some of that life science background. So by the end of the first semester, both cohorts are sort of matched in terms of their capabilities. And then the second semester and the third semester is where you really focus in on really advanced topics like process characterization and validation, quality by design, these high-level courses that distinguish you from some of your colleagues, your peer colleagues at other institutions. And then lastly, in your third semester, we have what are called industry-sponsored capstones, where you guys work on those projects that I told you about with the company as part of the final portion of your degree. And then we have here, this is just a sample of some of the concentrations that you have options. Actually, we added to this since I put this slide together. We also have a concentration that's starting in the fall, which is going to be focused on cell and gene therapy, which we're ramping up. And that'll be ready to go in the fall. So just some options here if you guys are interested. But I'll pause there because I know I give you guys a lot of information. You guys are a wonderful audience. And if you guys have any questions for me, just feel free to